Video number 5 for Sequencing and Introduction to Hail. For this video, video number 5, Analysis of Sequencing Data Using Hail, for the 2021 virtual workshop on Statistical Genetics Methods for Human Complex Traits, hosted by the Institute of Behavioral Genetics, University of Colorado Boulder. My name is Kumar Vurapan. The TAs for this module would be Tim Perturba, Carolyn Diaz, Dan Horrigan, and John Compitello. The learning objectives for this video would be to capture the needs for Hail in the analysis of genomic datasets. Well, to answer that learning objective, we'll be asking why use Hail, who are in the Hail team that makes this fantastic tool that is used almost throughout everything in genomics right now, what can you use Hail for? So to appreciate this better, Hail was developed by looking essentially at this plot here that was taken from the Broad Genomics group from Genomics in the Cloud, which is a book on Amazon that Geraldine Vandermeer wrote. So she's the engagement person I was mentioning in video. Well, I want to say the last video, 2 videos ago. Anyway, so genomics. We saw how genetics started in like 1980s with candidate genes, like tens and hundreds to millions of markers. But now that we've hit the 2010s, where we've reached the millions of markers, these millions of markers per individual is in gigabytes. Per datasets, if your datasets are thousands, like if you think about the Nomad dataset, which is probably like a petabyte, I would say, which is what, 1,000 terabytes, or anyway, a very, very large amount of data that is needed, or that is generated. And so from this data that's generated, the first issue would be, of course, storage, where storage, you can always find storage, especially using cloud systems, which tend to be also cheaper, by the way. Like at the Broad, we pay about $75 a terabyte on our Broad cluster, but if we were to do the same on the on Google Cloud, I think we pay about $25 per month. So like, and then the analysis on the cloud is also easier, but we'll have the next video talk about that a little bit more. But back to this plot here. The data generated from 2009, this one really shows an exponential increase in terms of amount of gigabases, and this is more than 10 petabytes worth of data that we're predicting by, well, 2025 really, or almost right now. And this is comprising of 230,000 exomes, 65,000 of the genomes, and 26,000 of the transcriptomes that make up all of this. The one thing, if you're probably wondering, why is it that genomes are far fewer than exomes, is just because exomes are cheaper to do, and in the early 2010s, there was a huge trend of essentially exome sequencing everything. If we go back to like the heritability, when we talk about heritability and chasing after missing heritability, the exomes were the try and maybe almost failed at some points, but somewhat also pointed us in very good directions forward. But we still have missing heritability, but that's like a whole different conversation altogether. Point is, this amount of data, the tools to analyze it is really not capable of taking this sheer amount of data, which is what Hail was developed in 2016, 2015, looking forward, the decade forward, of just like having the scalability issue being tackled before we even reach to that point, which is why Hail was developed, to take apart the analysis of these petabytes of data and making it easy. So this is a graph or a flowchart that we saw earlier of sequencing to FASTQ files to running the GATK that I went through with you, each of these phases, and subsequently having the variant call file, which is the VCF file that essentially can be read into Hail. So GATK is still used in order to take reads from the sequencer to form the VCF file that would then be used in Hail for your analytics. That would be used mainly in your statistical genetics pipelines. So the Hail team in itself is the team that created this, as a systems engineering team building tools to accelerate biological research, comprising of everyone on this slide. And I love this slide just to show you how we're a small team but has a really powerful tool that we use and share with everyone, well, in the genetics community. And we're also trying to make it as open to every other community outside of genetics that have big data questions that they'd like to solve, all led by Cotton, who is our, well, undying leader right now, and has two teams, which is the Curry team led by Tim Perturba and the Batch team that's led by Dan King. Two different parallels, which we will not get into in this workshop, but hopefully in the upcoming years, these nuances will be more projected. And it's made up of two non-software engineers, myself, who's a geneticist, and Whitney Wade, who is our operations person, who essentially keeps the head on wherever we're going. Where are we going even? So thank you. So these are all the people who are on our team right now, which make up HAIL. So HAIL, what is HAIL? In 2016, 2017, when we were at ASHG, before I joined the HAIL team, but I was part of the larger unit, many people asked what was HAIL, because it was more like, this is a tool, you can use it for multiple things, which is really what I do want to say, but at the same time, we do have some definitions. As a geneticist, I got you, boo. So Conrad, who was the lead analyst for Nomad and now is a PI in his own right, quoted this to us about 3 years ago. On a scale of 0 to dplyr, dplyr is a tool that is used very commonly in data science for R. The HAIL 0.2 interface scores an 8 out of 10 for general purpose data analysis. And I feel this is such a big compliment coming from Conrad, who deals with one of the largest data sets known to mankind for genetics in Nomad. So what is HAIL? HAIL is an open source data science library which allows you to slice, dice, query your data, and model any kind of data that you want, specifically in genetics. So if you think of it from the dplyr standpoint, it's more of a dplyr pandas as opposed to a plink, for instance. It's also highly scalable. That makes it really easy to use with both small data, which, you can still use Plink for small data that may work, small scale data, which may work really well. But for really large scale data that Plink breaks, this is when you use HAIL for biobank scale genomic data, especially for sequencing data. It's also a unified genomic data representation, which takes in every kind of genetic input, as long as it's tabular, like VCFs, BCFs, BGens, text files, just gene data sets, any kind of tabular data, and it unifies it into a form called the matrix table that is a single interface for working with all kinds of genomic data. And finally, a sense of community that may be blocked by my video over there, but I'll elaborate that we also have a huge community that is very active in the forum and chat room for people who are interested to think about further applications in HAIL, and also to ask us questions that you may have in HAIL. So most of the tools that you have are in HAIL, but we also like talking to you because we don't really know what you need until you tell us. We're not mind readers, So most of the tools that you need are in one place. And we also do like to remind everyone that we're very available to converse with you, not just about thinking of other problems that HAIL could undertake, but also if you have issues with maybe just like debugging something, or you feel like you want to think about can I use HAIL for my application? Come and let us know in our community forums and chat rooms. Honestly, our software engineers, especially Tim and John Compitello, who you will meet during our practical session, they are two of the people that you will answer for practically all of the things that you have questions for, which is great. So, and as I mentioned, we really cannot read your minds, so talk to us. Go on to discuss.hail.is and let us know what questions you have or what features you feel like we could encompass, or you've not read it in our documentation and you'd just like to be pointed towards it. Also fine. Don't be shy. We won't bite. So how has Hail been used? Using from our Hail is or hail.is website in the references page, I took all of the abstracts off of 2020 and just did a word cloud here, one of my favorite things to do. I'm a little bummed out that during this entire session I can't poll everyone that's here because polling is one of my favorite things to do using Poll Everywhere, for instance. So anyway, this would be how Hail has been using all of the abstracts. As we can see here, larger the words, the more it has been used, such as genes, variants, genetics, associations, disease, rare, which I really believe is possibly because of a lot of nomad it has empowered it, and multiple other keywords here which have encompassed a lot of things which are genetics related, which is where Hail is strong and most frequently used. So where has Hail been used? Just by looking at the reference page that I was mentioning, most of which are in the Northern American US region, some of which in the UK, where a large proportion are from Boston, which is not really surprising considering Hail was born at the Broad Institute, which is in Boston, Massachusetts, right over here in the northeast of the United States of America. And so that was not as representative as what I believe of the entire world, because I was shocked because I know of people outside of the US who have used it. So I just looked at everywhere that Hail has been pipped, which is a method that you use in Python that you would have probably learned from Cotton in your Python primer of where, aka Hail has been downloaded, which is truly a global area. And I've been very proud of the fact that we have also reached out to multiple countries in order to give Hail training, which is similar to what you're hearing today, except a little more compressed and, well, more time to debug on your Hail front. So if you are more interested in using Hail and you feel like you want a workshop by itself, reach out to me at verapen@broadinstitute.org or on Slack, and I can arrange for that. But the point of this plot here, looking at the map, is just to show you that Hail has been used in almost every continent on Earth right now, showing the ambiguity or showing just the sheer use over global use of Hail right now. And also, sorry, I didn't make this other point. I can't make it, but if you go back to your slides, the darker the blue, the more Hail has been pipped, and the less dark the blue, the less Hail has been pipped. So all of that is adjusted as well to the population size. So back to now what our conversation is, why would you use Hail? So when we think about a lot of genetics tools, implementing those tools on the cluster or on the cloud will take so long trying to debug it. On top of that, sometimes trying to wait for it also takes really long. And you don't really have a lot of time to reason your science, because that's really what we're here for, to come up with scientific reasoning, to figure out what the results tell us, and also from there, do we need to do more experiments? And by the time you reach there, you have to redo some of the implementation if you need to use new tools, and you have to wait if you're using the old tools, or after implementing those new tools. There's a whole bunch of things that you have to think about. So Hail wants to try and make the implementation shorter, the runtime shorter, just because of the scalability that Hail provides. And to share with you one example of what I went through in 2016, which was when I first joined the lab for Arnaud Palotier. We worked with 4,000 whole genomes at that point. This is almost a terabyte worth of data. So running the analysis for it was a pain if you were to think of using a cluster-based analysis tool. On top of that, I also remember running VEP, which is an annotation tool to annotate each of the variants and what it means. It usually will take forever using cluster-based tools. So with Hail, VEP, using about, I want to say, 60 cluster nodes, which is kind of like 60 computers on the cloud, took about an hour, which I think is really fast for 4,000 genomes. Another thing that I think about is also the QC steps. Just running the QC after I've already established what QC parameters that I want to filter down for variants which are of interest or at least of high quality at that point. Wow, it took me probably in total, about 15 or 20 minutes for 4,000 genomes. And this was Hail 0.1 in 2016. I've tried implementing this in Hail 0.2 just for shits and giggles, just to see how it works and how fast it is. It is probably two times, if not four times faster than my code running with Hail 0.1. So I had more time for scientific reasoning, and the implementation and runtime was almost close to nothing because an hour and 30 minutes of my time is nothing compared to before, which took almost like a week or two to run all of these steps in my pipeline. So Hail, as a data science library, we have like two sections here, which is the data slinging and the analytical toolbox. So when it comes to data slinging of how you would run your data or just like import your data, which will be read and writing your data, you could read from VCFs, TSVs, BGENs, plinks, JSON files, GEN files, which is a non-binary version of BGEN, BED files, which is a plink file, and GTF file, which is something you would get from RNA sequencing outputs. Point is, anything that is tabular can be essentially read into Hail as a matrix table. And the matrix table is the part that unifies all of these different file formats, which I know you're just like wondering, another file format. No, because the concept that we're trying to push right here is you have all of these other file formats that you're just importing in, and then you just have that one file format that one works really fast in Hail, and secondly, when you export it out, is also much tinier than the starting material. An example of this would be, for instance, Nomad, which is, I think, 140,000 whole genomes right now, version 3. So in Nomad version 3, they only export all of their data in matrix tables and no longer in VCS, just because VCS are much larger and less portable compared to matrix table. I'm not saying that matrix tables are not large, depending on the number of exomes and genomes that you have. So from there, you can also do filtration, grouping, and aggregation of your data, the slicing and dicing of the data to compute, for instance, mean depth per variant per sample among the heterozygotes, and then you can also group them by ancestry labels and sex. You're also able to, like another example here, to count transitions and transversions called per sample. And I did also allude to the VEP tool, which is the variant effect predictor tool by Ensemble. Now we even have a setup that you can just VEP with a single line without even downloading VEP onto your local directory or onto your virtual machine on the cloud. So you can just use a single line, which I think is, it is hl.vep. Put in your matrix table in that command, and you would annotate immediately with all the VP. I will not be demonstrating this during your practicals, but I'm happy to walk you through some of this and also point you to some of the resources on Hail if you want. This is one of my most excitable tool, if you will, other than the VCF combiner in video number 3, just because, VEP was such a pain for me in grad school. So then you could also join your annotations by variance, local or interval, locus or interval with genes. You really get to choose how you want to slice and dice your data. Think about it as dplyr, except Python-based, and you could use it on the cloud, which is where it works really best. And you could also use our annotation database to then develop other kinds of annotations to annotate your genomes or exomes or transcriptomes out. Finally, visualization uses bokeh from Python in order to produce visualization. I personally prefer still visualizing using ggplot2. So what I usually do is actually export the data, but for the interest of just like you learning right now, we also do provide visualization where you can just visualize your data and import the PNG or JPEG files in order to just look through them. They're also all publication ready, which also makes it really easy for you to just go on with your life. So the analytical toolboxes, we have many statistical tools for genetics, and the one thing that we use very frequently that I'll be demonstrating is the linear regression method that has been used with the UK Biobank Rapid GWAS, which is the GWAS that was run by Ben Neal's lab of $1 a trait, which I still think is fantastic because $1 a trait on the cloud, amazing. And we do have linear regression tools, which are still big for the most part. But before we go on to that, my apologies. We also can run PCA using PC Relate on Hail. And this was PCA from one of the previous versions of Nomad. And I was also mentioning we do have some linear regression tools, such as Inferring LD, shown here in UK Biobank, which is one of our first tools for linear algebra applications. We do have many others, which are mostly in early stages, I would say mid stages right now, led by John Compitello. And if you have questions, again, feel free to ask us on Slack. John is on it, and he's happy to take questions for the linear algebra questions. So now let's backtrack a little towards the matrix table itself. So I did mention there were multiple formats that Hale can take in and unify. One of the formats that I talked about in our previous video is the VCF format, called the variant call format, or the varcol file, if you will. In this file, it's a tabular file where the column IDs are the sample IDs, the variants are the rows, and the intersect would be the genotype, the allelic depth, and the depth itself. In rare variant aggregations where you don't have the input file format as per variant, unlike the previous slide, now would be a per gene because you're collapsing the variants, and you'll be learning more about rare variant aggregation and other kinds of tools, especially with Sage Gene, in your next module with Wei Zhou or Zhou Wei, of how I would prefer to call her because it's the proper way of calling her. Anyway, point is, here, for rare variant aggregation, the column IDs would still be IDs, and the rows now would be genes instead of the variants, and the intersects of them would be the how many loss of functions, number of missenses, number of synonymous, and sometimes you'll also have p-values if you've already run your analysis and just running it in Hale to just observe certain things. But, this would be the raw file that you don't really have p-values. You also can put in trio data, where similarly the trios would be at the top. The variants now would be the rows, and the intersects would be the genotypes of each of these proband mother-father genotypes. Transcript expression, which would be for RNA-seq data. The columns would be the tissue samples, the rows would be the transcripts, and the expression would be what you are getting in between the intersect of the sample and tissue. So all of these file formats can be imported in as a matrix table, because a matrix table has columns and rows, and the intersect would be entries where the corresponding rows table that stores all the rows fields is constant for every column in the dataset, and a corresponding columns table that stores all the columns field that are constant for every row in the dataset. This is really two-dimensional because of the column row intersect, which make it really easy to be indexed and called upon. So the index is usually by rows and by columns, so upon indexing you would potentially have many fields for you to easily call and pick upon every time you need it. So the set of the global fields here are constant for every entry in the data set, and I'd like to show you this example. So this example is the output from just looking at the matrix table, where the global fields here, you don't really see it as entries, instead you see it later on. So the column fields would be the samples, the row fields would be the locus, the alleles, the RSIDs, the identifiers genetically, and the entry fields, which at this point you will see it as almost exactly similar, if not the same, to what you see in the VCF. The difference now lies in the fact that, other than the fact that these are intersects of the columns and the rows, you are now also putting in a key of rows by locus and alleles, and key by samples, which make the intersects very easily to call upon, and this is why it makes really fast slicing and dicing of your data when it comes to it. Another thing that you also see is the Hale table, where the Hale table is just a two-field group, which is just the rows and the global fields. This is usually things like annotation data sets that you'd want to put in and just like join with your matrix table. And I promise you that when we reach the practical, I will show you the difference between the matrix table and the table, and how you would join your own annotations and not just VP into your matrix table. We also have pretty cool cheat sheets that John came up with, which is on this website. It's very clickable when you have the slide deck. And it's one of my favorite things to show people because it also shows you quick and easy commands in order to slice and dice your data easily. So mastering Hale takes a lot of practice. And it's, well, I would say it's relative. If you are very comfortable with programming, it's much faster. But if you've already learned, simple shell script, this may be a little higher or steeper on the learning curve. It is harder to learn compared to most command line tools because it's not just about memorizing command line calls, because you'd have to understand the foundation of data science skills in order to use the commands that would allow you to slice and dice your data. Prior experience with a data frame library such as R, dplyr in R, or pandas in Python can be extremely helpful into learning how to use Hail. Believe me, as a person who did not major in computer science or even statistics, learning R and dplyr to begin with makes Hail so much easier on the learning curve. Hail is truly about giving you the tools that you need to indulge. Just indulge your time, like having a piece of chocolate from Godiva and you're taking your time. This would be what Hail would be for your scientific curiosity on biological data. And that's really not truly always easy. And Hail provides you, and we feel confident that we are. Hail can be used for a lot of large-scale data, such as these data sets here in UK Biobank, which, 500K, which can grow up to 5 million for almost any other biobanks out there, like all of us, which is 1 million, FinGen, which is up to 500,000 future iterations, and Nomad, which is now almost 150,000 whole genomes, amongst all of these other data sets. So instead of from bringing data to researchers, we're bringing researchers to the data. So right now, we're bringing you to the data as opposed to getting someone else to churn the data for you. You can do it yourself because these tools can be easy. I did mention that the learning curve may be steep, but believe me, after you learn how to run Hail, your life is so easy to do many things that you don't really even have to think about, like, do I have time to go spend with my parents or my children or my grandchildren? Instead, you'll now be thinking with Hail, I have all of this extra time. Let's go for a hike. Let's go shopping, which is what I keep telling my mom. Let's go shopping. Does she enjoy it? Yes. Pandemic? Not really easy because online shopping, not the same. So Hail allows you to use in multiple computational landscapes, such as locally in your local or laptop or desktop, server, high-performance computing, or cloud. So in your laptop is mostly for developing your Hail scripts and commands. It's usually for very small data sets, like tens of genomes or hundreds of exomes, if you will, depending on the amount of RAM that you have in your computer. For servers, like own servers that you have in multiple computers, just like in your lab, for instance, medium scale data of about thousands of genomes to tens of thousands of exomes. You can use this on high-performance computing clusters for large data sets of 1 million genomes or even tens of millions of exomes, and cloud would be similar. The caveat that I would have is we don't really support high-performance computing clusters. So this slide here shows you how you could get started, which is just essentially doing pip install Hail on your command line. And this is a Python tool that allows you to install Python packages on the fly, and Hail is an index Python package. Python library. I use R a little too much. So using Hail as a Python library, you can download it onto your laptop or desktop server as well, or a single node HPC that you can use like on Slurm, Ugar, or LSF. Again, I'd like to reiterate, we do not support HPC schedulers, just because most, if not all, of our support is on cloud-based tools, where the most commonly used one, especially at the Broad, is GCP, Google Cloud Platform. Even your practicals are based off of Google Cloud Platform that you can get started by using pip install Hail. And if you were to run a cluster by itself, you could use Hail control or Hail CTL, dataproc start, and name your cluster all in small letters. You can also use Amazon Web Services. We do have some support, but I would not even call it like super big support as opposed to GCP. We do have like Garrett Bromley, who does come on board every once in a while to help our users with AWS if you do need to debug your AWS code for Hail. So what would be your next steps? To summarize, really, pip install Hail is one of your like first lines of code. Secondly, read the documentations. We've written as many documentation pages as we have. If you have any suggestions on how to make it easier or how to make, if something was confusing for you, feel free to reach out to me personally and I'll take care of that. So the website address is hail.is/docs/0.2. And you can also go onto our blogs. We have 2 blog posts right now. And our blog post is not really in a high cadence just because most of our blog like resources have gone to COVID-19, the Host Genetics Initiative. So go to our blog post to see an introduction onto Hail. There's some pretty cool snippets there that I find very interesting to use and useful. And finally, the most important place to go to is discuss.hail.is, because if these 2 things don't pan out and you feel like you are at a wall, know that we're here for you. And I can't reiterate this enough that our software engineers do check discuss.hail.is almost in different time zones, even sometimes if you look at it, where the turnover rate is almost like right now we're targeting like a 1 hour turnover rate upon you posting something on the Discuss. So the learning outcome for this video would be Hail is a useful tool for analyzing genomic data, as I've shown you for multiple tools, multiple input formats, and how you can analyze your data, especially for large data sets by using the matrix table. The Hail team comprises of fabulous people, obviously. Thirdly, Hail can be used for almost every genomic and especially sequencing-based questions that you have. And the learning objectives for the practical sessions that I will go through with you and just mention at the point is to be able to use basic Hail functions. I do not expect you to be experts at Hail, but just knowing the introductions. And on top of that, after we're done, I'll be posting all of the code on my GitHub repo so that you can just use the Docker image to run it yourself, either locally or on a cloud to set up. You can also use it to apply basic GWAS analysis tools using Hail on your own datasets, to describe the use of PCA in Hail to decipher ancestries, to obtain resources in order to explore further the extent of Hail capabilities that I've already shared. Session that we meet on June 16th. So this brings us to the end of this particular video. I will see you on June 16th. We do have a bonus video, which is the 6th video on using the cloud and how Hail can be used on the cloud to empower your analytics. My name is Kumar Veerapan. Feel free to reach out to me on Slack, on the IBG Slack, or even send me an email at veerapan@broadinstitute.org. Thank you very much.